The plasma membrane. The plasma membrane is a biological barrier that separates the interior of a cell from its outside environment. The fundamental structure of this membrane is the phospholipid bilayer, which forms a stable barrier between two aqueous compartments. The structure of the bilayer is two layers of phospholipids back to back. A phospholipid is comprised of its lipid tail, which is nonpolar and hydrophobic, and its phosphate head, which is polar and hydrophilic. This means that the membrane is impermeable to ions on most water-soluble molecules. Whilst the bilayer is integral to the membrane structure, it is not its only component. Cholesterol plays a role in the membrane's fluidity, but its most important function is in reducing the permeability of cell membrane. It fits in between phospholipids and prevents water-soluble molecules from diffusing across the membrane. Proteins also play a major role in the membrane. They're used for cell recognition as vessels facilitating diffusion and maintain the membrane's fluidity. These can be either intrinsic or extrinsic. With intrinsic proteins, they span the entire membrane, meaning they cross both sides. This is not true for extrinsic. Carbohydrates are also present within the membrane, forming glycolipids and glycoproteins on the extracellular side of the membrane. Glycoproteins and lipids act as recognition sites, as well as anchors that provide structures to the membrane. The many molecules that form the membrane give it its fluid mosaic properties. The fluidity of the membrane arises from the phospholipid's fluid nature. Movement of the membrane means it is never completely impermeable. Due to these many structures within the membrane, molecules of many sizes can enter one way or another.